Hello, everyone, and welcome to our webinar, Rethinking Therapeutic Response Assessment in Clinical Trials Using Ultrasensitive ctDNA. We're excited to highlight some key data on utilization of ctDNA and lymphoma today, including some data that was recently presented at the American Society of Hematology meeting this past December. We're very excited to have some expert panelists with us today, including Dr. David Kurtz, Assistant Professor of Medicine at Stanford University and co-founder of Foresight Diagnostics, Dr. Ryan Lynch, Associate Professor at the University of Washington School of Medicine and Clinical Research Division at Fred Hutchinson Cancer Center, and Lara Stepan, Senior Principal Translational Research Scientist at Cancer Immunology and Cell Therapy Thematic Research Center at Bristol Myers Squibb. In her role, she supports cellular therapy programs. I'm going to begin today with a brief overview of ctDNA assays and some considerations around assay performance before we shift into some data rounds with our panelists. We will have time at the end to answer any audience questions. Before we kick things off, I want to specify that the information provided in this webinar is for educational purposes and has not been evaluated by the FDA. The products featured are research use only and are not intended for diagnostic procedures. Right. Circulating tumor DNA-based MRD detection is poised to address some critical clinical limitations in the lymphoma treatment paradigm of today. PET-CT imaging continues as the standard of care to detect relapse and refractory disease, despite well-known limitations in its performance. And although there is a lot of very exciting therapeutic development in the lymphoma space, guidance for therapeutic decision-making is limited by the absence of actionable biomarkers to identify patients who are most likely to benefit from that therapy. Although MRD testing may address these issues by improving disease detection and guiding therapeutic selection, we are facing several research challenges on the pathway to making that a reality. Traditional clinical trials with outcomes-based endpoints take a long time to read out and assess the efficacy of new therapies or the use of biomarker-based therapy selection. While ctDNA may accelerate trial readout, the use of molecular biomarker-based primary endpoints does have limited precedent. The MRD space more broadly is also at a crossroads, with mixed performance of MRD tests and other indications being reported recently. Results from the Galaxy trial were shared at ASCO GI in January, where investigators demonstrated the prognostic performance of MRD testing in stage 1 through 4 colorectal cancer, where patients who were ctDNA positive post-surgery had worse outcomes than those who were ctDNA negative. At the same conference, data was also presented from the COBRA trial, where MRD-informed therapy selection was ineffective in resected stage 2 colon cancer, and ctDNA clearance was actually worse with MRD-guided treatment compared to the observation arm. These opposing findings in trials utilizing different MRD tests have triggered discussion of if and how to incorporate MRD testing in colon cancer. This very recent example has also highlighted that although ctDNA detection has the potential to improve clinical care in oncology, not all MRD assays are created equal. Many different technologies have been developed to detect ctDNA to date. However, the specific technology being used does have downstream impacts on assay performance and potential utilization scenarios. We've highlighted here some of the key differences amongst assays, along with the strengths and weaknesses of those approaches. Most ctDNA MRD assays are based on DNA mutations, while some approaches do consider DNA methylation. Assays can also be tumor naive, meaning that the same mutation or methylation signature is evaluated for all patients within a cancer type, or assays can be tumor informed, meaning that a patient-specific tumor signature is used to detect ctDNA. Assays can utilize off-the-shelf fixed panels, meaning that the same genomic regions are assessed in all patients to develop a tumor signature, or they can be custom, meaning that even the genomic regions being interrogated are patient-specific. The combination of these features can impact the clinical performance and applications of an assay, making it critically important that MRD assays not be lumped together. Decisions about whether a tumor-naive or tumor-informed approach is utilized may be influenced by sample availability or other logistic factors, but it does have a direct impact on assay sensitivity. It's also important to pay attention to what metric is being used to report on assay performance, with LOD 95, or the limit of detection that can be achieved with 95% probability, being the standard performance metric. The LOD95 is distinct from the lowest limit of detection achieved for an assay, which might be reported as a limit of detection as low as fill in the blank. This typically represents the lowest ever detected sample for an assay, which might not reflect the assay performance in a typical case. The LOD95, on the other hand, is representative of overall assay performance and what can be expected in a trial or clinical setting. It's also important to consider the assay requirements to achieve a certain LOD95. If hundreds of micrograms of DNA are required, it may not be feasible to achieve the reported performance in a real-world setting. Considerations of the application for ctDNA detection should really guide assay selection in these features. For things like early detection, it's necessary to trade off performance for a tumor-naive approach. However, the assay sensitivity achieved with tumor-informed approaches is critical for ctDNA detection in conditions or at time points with very low tumor burden, like at the end of therapy, or to significantly shorten the time for detection of disease recurrence. And even within tumor-informed approaches, the specific approach can significantly impact test performance. And these low tumor burden applications, reducing background noise is critical to achieving the low LOD95 needed for ultrasensitive detection. At Foresight, we utilize the phased-seq technology developed by our co-founders at Stanford. Phased-seq is a tumor-informed approach that leverages the presence of phased variants to detect ctDNA. Phased variants are two or more single nucleotide variants, or SNVs, that occur in close proximity on the same DNA molecule. This allows the background error rate to be substantially reduced for phased variant detection relative to SNV detection-based methods. The LOD95 for our assay is in the parts per million, providing an ultra-sensitive approach for ctDNA detection. The assay leverages the mutational hotspots observed in B-cell malignancies to use an off-the-shelf panel for lymphoma, but utilizes a custom panel for solid tumor cancers where mutations are more distributed across the genome. Data rounds today will highlight the use of phased-seq testing for MRD detection. Dave Kurtz will kick things off with a discussion of optimizing assay sensitivity for accurate response assessment and prognostic performance. Then Lara Stepan will discuss the use of ctDNA as an early outcome predictor for lysocell in the TRANSFORM study. And Ryan Lynch will close things out with an evaluation of the most promising near-term clinical applications of MRD assessment. And with that, I will hand things over to Dave.
All right. Thanks for that great introduction, Crystal. I think that was a fantastic overview of what we want to talk about today during this webinar. So my task today is to go over some of the recent data that's been reported from our collaborators at Foresight at the most recent ASH annual meeting, as well as some additional data presented at AACR and emerging data that's come out of Foresight and our collaborators. So I will not be able to go over every abstract that was presented at ASH, but I think there were a few key highlights and themes that emerged from the ASH annual meeting. The first of these is that MRD measurements are potentially more prognostic than PET-CT scans during and particularly at the end of therapy for first-line large B-cell lymphoma. Second, using ultrasensitive ctDNA measurements with phase-seq enables detection of residual and recurring disease 12 months earlier than using PET-CT PFS-based alone, potentially unlocking novel clinical trial endpoints. And finally, achieving an LOD95 in the parts per million range can improve prediction of clinical relapse across multiple time points. This is from pre-treatment, again, particularly going through the end of therapy for large B-cell lymphoma. So with that in mind, I want to highlight this presentation from Jordan Goldstein as the first author from the ASH annual meeting, looking at the analytic sensitivity that can lead to improved clinical sensitivity across milestones in first-line therapy for large B-cell lymphoma. So here we define clinical sensitivity as the fraction of patients that are detectable who will ultimately progress within 24 months of their large B-cell lymphoma therapy. So here we took a pooled analysis of patients from prior studies using the phasing technology and assessed how well different thresholds could detect residual disease at various milestones for large B-cell lymphoma. This includes prior to therapy, at early time points during therapy, and then ultimately at the end of therapy. And what you can see is that while detection of ctDNA in large B-cell lymphoma prior to therapy is relatively robust across various analytical sensitivities and LODs, as you progress through treatment, a higher and higher limit of detection is required to robustly detect disease. And this is, again, most significantly noted at the end of therapy, where having a limit of detection in the parts per million range enables detection of the vast majority of patients who will ultimately go on and have a clinical disease progression. How does this look in the real world when we compare this to standard radiographic imaging? This is one example of that, highlighted by Mark Rashowski at the NCI, looking at their study of the combination of acalabrutinib plus standard first-line anthracycline-based therapy in large B-cell lymphoma. Similar results were shown in other presentations, but I think that this result really highlights how stratification using ctDNA at the end of therapy can outperform stratification using standard radiographic responses. So on the left, we can see stratification by end of therapy PET CT scan as assessed by blinded radiologists reading that end of therapy scan. You can see modest stratification in this cohort using patients that had achieved either a PET CR or did not achieve a PET CR at the end of therapy landmark. And importantly, in patients that did not achieve a PET CR at the end of therapy, the majority of these patients do not go on and have disease progression within the first 24 months after therapy. Now, in contrast, when we look at the performance of ctDNA MRD performed by PhaseSeq at the end of therapy, we see much more robust stratification, higher hazard ratio, and again, importantly, the patients that are positive for ctDNA at the end of therapy overwhelmingly go on and have their disease progress, have a clinically meaningful event, showing the increased specificity of this ultrasensitive approach. So this is something that we've seen at Foresight using PhaseSeq, not just in lymphomas, but in solid tumor studies as well. So this is reported by some of our collaborators at Sloan Kettering, led by Jay Isbell, looking at a cohort of patients with early stage non-small cell lung cancer, looking at landmark detection, so a single time point for detecting residual disease after curative intent surgery for non-small cell lung cancer. And again, using the Foresight custom MRD assay based on PhaseSeq, we see robust stratification of outcomes, where patients that do not have residual disease, as measured by PhaseSeq, do very well, whereas patients that have residual disease go on and have disease progression. Now, importantly, most of the patients that have detectable disease actually have disease detection below 1 part per 10,000. In contrast, when we compare this head-to-head in the same samples using an SNV-based tumor-informed detection method, we see, again, stratification in outcomes using the less sensitive approach, but we see that this stratification is much more modest, and essentially we wind up missing patients who truly have residual tumor burden, but just don't have detectable disease at the limits of detection that are required to detect the low burden of disease at this critical post-surgery and post-treatment landmark. Now, I want to finish with the last few minutes with a couple questions. The first of these is looking at a use case. What can we really do with ultrasensitive MRD? The first use case is with the concept of accelerating clinical trials with earlier endpoint readouts. And this is really borrowing a concept for what we're calling EFS with MRD. People have previously called this modified progression-free survival using radiographic imaging. And really what this is taking a standard PFS definition, which for large B-cell lymphoma would be relapse or progression of DLBCL at any time after initiation of treatment or death from any cause, and adding one additional type of event. And this is a single measurement with the detectable residual ctDNA after completion of therapy. And to include this time point, we really require high sensitivity and specificity to try to capture as many patients as possible who ultimately have residual disease. In the Kaplan-Meier plot on the right, you can see three candidate endpoints for a possible clinical trial. This includes overall survival, progression-free survival, and EFS with MRD at ultrasensitive limits of detection. And what you can see when you compare the red versus the green curves is that ultimately the plateau for PFS and EFS with MRD are very similar, but you get to the events in the green curve much, much more quickly. We can see this more specifically when we consider a hypothetical clinical trial where we try to have a target event rate of, let's say, 25%. If you considered OS as your primary endpoint, within 24 months, you would not have achieved a readout of this trial. PFS in this cohort of patients would have read out the trial at 18 months. But if you look at EFS with MRD, with ultrasensitive limits of detection, we can see that we read this trial out a full 12 months earlier than if we used standard progression-free survival. Now, importantly, this really only works if you're classifying patients properly. So you want to make sure that PFS and EFS with an MRD event are essentially capturing the same information, that they're giving you the same patients. And you can see this on the right with this alluvial plot, where 95% of patients, when you compare the PFS endpoint versus EFS with MRD endpoint, are having the same classification. You just get to that classification a full 12 months earlier. So that's as shown here as a 95% concordance with a traditional PFS endpoint. A second potential use case is actually trying to use this ultrasensitive MRD assay to enroll patients onto a clinical trial. And this is particularly highlighted with the announcement of Allogene's ALPHA3 study, where we're asking the simple question, for the patients that have detectable MRD at the end of therapy, this population and the red curve shown here, what can we do for them? How can we improve their outcomes? So this is a partnership between Allogene Therapeutics and Foresight Diagnostics that was announced just in January of this year, where Allogene is taking a really novel approach to try to intervene on these patients. So using the Foresight PhasED MRD platform to identify patients who have the lowest burden of residual disease left after therapy, detectable MRD by PhasED, and then giving patients Chemicell, an allogeneic CAR T cell, to try to improve outcomes for this patient population. So in conclusion, from this section, I've highlighted a number of things, but I think that we've shown that the diagnostic performance at the most difficult and clinically meaningful time points when disease burden is low require ultrasensitivity for an MRD platform. This includes at the end of therapy for first-line DLBCL or after surgery for a curative intent non-small cell lung cancer. Standard of care response assessment, which is currently done via PET-CT scan, can potentially be improved using ctDNA MRD. And finally, clinical trial execution and success using MRD as either a potential endpoint to accelerate clinical trials or as a biomarker to identify high-risk patients for consolidation approaches are emerging methods that we can take forward into clinical development. Thank you very much and look forward to the questions.
All right, for my data round today, I will be sharing ctDNA data from the Transform study that was first disclosed at the ASH Symposium this past December. Our Transform data will be showing ctDNA dynamics is associated with clinical outcome and that achieving undetectable ctDNA status is an early outcome predictor following lysocell treatment in the second-line DLBCL study. I'm going to start out with providing some background information to help put our data into context. Our Transform study is BMS's phase 3 registrational study in high-risk second-line DLBCL patients. These patients are refractory or relapsed within the first 12 months after frontline treatment. Patients are enrolled into one of two arms for the study. They are randomized into either the lysocell arm or the standard of care arm. Patients randomized into the lysocell arm receive lymphodepletion prior to lysocell infusion. Bridging therapy is allowed. I want to highlight that lysocell is our Breyanzi cellular product. It is our CD19 CAR T cell that is approved for second-line plus DLBCL. Patients that can also be rolled at a 1-to-1 randomization rate into the standard of care arm. The intended treatment for standard of care is to receive 3 cycles of platinum-based standard of care, followed by high-dose chemotherapy and transplant. Patients enrolled in the standard of care arm can roll over to receive lysocell treatment based on an EFS event. The key goal of our Transform study is to show superior efficacy with lysocell treatment over that of standard of care. The primary endpoint for our Transform study is event-free survival, and our primary analysis results demonstrated superior EFS in the lysocell arm over the standard of care arm. Median EFS was not reached for lysocell patients versus 2.4 months for standard of care patients, with a median follow-up of 17.5 months. In a few slides, I will share data from our lysocell patients showing that ctDNA clearance is highly correlated with EFS. For our exploratory analysis, we used Foresight's ultrasensitive phased-seq ctDNA MRD assay. We used pre-treatment plasma samples and matching germline DNA to identify phased variants to develop the ctDNA MRD monitoring assay. No tumor specimens were required. We performed assay calibration using baseline samples for all patients enrolled in the study, patients in the lysocell arm and in the standard of care arm. Phase variant identification and assay calibration was successful for the majority of the patients. There was only a 15% assay calibration failure rate, and the majority of those calibration failures were associated with low SPD tumor burden. I'm not going to show this data today, but pre-treatment ctDNA levels are correlated with measures of tumor burden, SPD and LDH, and they are also correlated with high-risk characteristics for the patients, IPI score and Ann Arbor stage. So while we performed assay calibration for all patients enrolled in the study, lysocell patients and standard of care patients using baseline samples, we only assessed longitudinal samples from patients that received lysocell therapy. Post-lysocell effusion, we assessed various time points ranging from day 15 to month 12. Our goal is to understand the prognostic value of ctDNA dynamics after lysocell treatment. So looking at our ctDNA kinetics, our data shows that ctDNA changes post-lysocell infusion track with response calls. We can see this looking at this plot where ctDNA concentrations are plotted by PET-CT response at month 3. Month 3 patients with complete response in purple rapidly and significantly reduce ctDNA levels. You can see this as early as day 15. On the other hand, progressive disease patients in black maintain persistently high levels of ctDNA. In progressive disease patients, we see no real depth of reduction. Next, we wanted to understand the correlation of achieving ctDNA negativity with clinical response. Here we show ctDNA negativity plotted by PET-CT response at month 3. And our data shows that month 3 complete responders achieve ctDNA negativity, with 50% of the complete responders achieving ctDNA negativity at day 15. And this molecular response continues to deepen over time, ultimately reaching 86% at month 12. This molecular response for the complete responders is also durable. Very few complete responders revert back to ctDNA positivity. On the other hand, progressive disease patients in the black line tend to maintain consistently positive ctDNA levels. So achieving ctDNA negativity is associated with complete response. Here we show achieving undetectable ctDNA status in the first 3 months after lysocell infusion is important and correlates with durable event-free survival. Our data plots here show that patients with undetectable ctDNA in the blue line have an EFS benefit over patients with detectable ctDNA. You can see this as early as day 15 with a hazard ratio of 3.26. And you can see this more significantly at month 3 with a hazard ratio of 8.16. These patients are in both radiographic and molecular remission. This data highlights the value of ctDNA as an early outcome predictor. On this last data slide, we focus on complete responders. Complete responders as measured by PET-CT that are still positive for ctDNA. So our data shows that ctDNA positivity correlates with worse EFS outcome. ctDNA positivity is a biomarker for progressive disease risk. This is demonstrated early at month 1 with a hazard ratio of 4 and more strongly at month 12 with a hazard ratio of 24. All 4 month 12 complete responders that are still positive for ctDNA go on to experience an EFS event. This data highlights the prognostic value of ctDNA profiling beyond PET-CT. So to summarize, I have shared ctDNA data from the TRANSFORM study in the second-line LBCL patients that have received lysocell treatment, and our data shows that lysocell induces a rapid, deep, and durable ctDNA molecular response that is strongly associated with event-free survival. These results demonstrate the value of MRD monitoring for disease surveillance and early prediction of clinical outcome in a lymphoma setting. Furthermore, these results highlight the promise of ctDNA MRD endpoints as an early indicator of therapeutic response, which could potentially accelerate study timelines. And with that, I'd like to acknowledge and thank all of my amazing co-authors and partners, both at BMS and Foresight. Thank you.
Thank you so much. So I will be wrapping up this morning's webinar. My name is Ryan Lynch again at the University of Washington School of Medicine. And so I will, the final handful of slides are going to talk about what are some potential clinical utilities of MRD assessment in various tumor types. Okay. So PET-CT in lymphomas is the standard tool for response assessment, typically utilizing the Lugano criteria, which are now about a decade old. And so there are some performance gaps with PET-CT, and so these can lead to challenges in determining patients who are truly in remission or patients who truly may be experiencing disease progression. So one of the largest data sets came from the Goya study, which was a phase 3 randomized study of 2 different CD20 antibodies in untreated large cell lymphoma. And in this data set, all patients received PET-CT at the end of treatment. And before that, many large cell studies did not consistently collect PET-CT data. So in this data set, the PET-CT, the positive predictive value of PET-CT for eventual relapse was less than 50%. And for patients achieving a complete response, the negative predictive value was less than 85%. And so this calculates out to a clinical sensitivity for detecting residual disease of around 30%. Our site participated in the multi-center review of large cell lymphoma patients analyzed with phase-seq at the end of treatment. That was led by Mark Rashofsky at the NCI. And when we looked at our data set of over 100 patients, the positive predictive value of detectable ctDNA at the end of treatment exceeded 80%. And strikingly, the negative predictive value was over 95%, leading to a clinical sensitivity of 90%. And so additional comparisons of ctDNA for end of treatment response assessment versus PET-CT have also been presented most recently at the ASH meeting last December. So there was a cohort of prospectively collected patients with large cell lymphoma at Samsung Hospital in South Korea who had ctDNA measured at the end of treatment, as well as paired PET-CTs. And so while PET-CT is prognostic for both progression-free survival and overall survival, ctDNA appeared to outperform both of those in this data set with much higher hazard ratios and compared to PET-CT. And this did actually remain statistically significant with a multivariate analysis of various baseline factors. And when we combine both that Samsung Hospital data set as well as our NCI data set, just looking at patients who have discordant PET-CT results and ctDNA at the end of therapy, we found that patients who were ctDNA negative but PET positive, none of those patients eventually relapsed. And in contrast, most patients who were PET negative but MRD positive eventually relapsed within 2 years. Some of the issues with PET response assessment also transitioned over to classical Hodgkin lymphoma. PET-CT is also widely used, and there are actually many PET-adapted approaches which are widely utilized in Hodgkin lymphoma. However, in recent years, the addition of checkpoint inhibitors have led to challenges in utilizing PET-CT in response assessment. And this is both in PD-1 inhibitor monotherapy as well as combinations. And so this just highlights a single case that was published looking at a PET scan in a patient before starting pembrolizumab, a checkpoint inhibitor, with their burden of disease. And then after the pembrolizumab, the disease appeared to worsen, but then with no additional change in therapy, the patient eventually achieved a complete metabolic response. And so many patients may have what we call pseudoprogression or just immune flare at the sites of treated lymphoma, and this can be hard to distinguish on a PET-CT in real time. There are a couple different studies that were presented at ASH or updated at ASH looking at frontline use of PD-1 inhibitors and chemotherapy and the performance of PET. Our center led a 50-patient pembrolizumab and AVD concurrently treated frontline study, and many patients had residual PET positivity at the completion of treatment. And this, so in our study, out of those 50 patients, about 23% had some level of FDG uptake after all 6 months of treatment. And this is despite having a 2-year PFS of documented biopsy-proven progression of only 97%. There was also a multi-center phase 2 study called the Keynote C11 study that gave patients 3 cycles of pembrolizumab, followed by a PET-CT, then additional chemotherapy, followed by another PET-CT. And I think what's striking about this is that compared to historical controls of chemotherapy alone, our PET negativity rates at the equivalent time points appear much less. And this is despite the fact that very few patients have progressed on this study so far. So as part of a collaboration with Stanford as part of an NIH funded grant, we actually looked at our first 29 patients treated with pembrolizumab and AVD and looked at both metabolic tumor volume and correlated that with ctDNA levels. And so in the top figure, we actually found that the ctDNA levels at baseline appeared to correlate with the total metabolic tumor volume, and that we could actually stratify early stage patients and advanced stage patients based on their ctDNA levels. And the waterfall plots on the figure below look at the reduction, the log change in ctDNA from baseline in all patients analyzed on this study. Strikingly, only one patient has relapsed so far in this study, and that's indicated by the red bar. So most patients by even cycle 3, day 1, clear their ctDNA, about 80% of patients, and virtually all cleared by the end of treatment. But indicated by the squares at the bottom, many patients retain residual FTG uptake on PET, despite extended follow-up not demonstrating any evidence of disease recurrence. So right now, where are we at in terms of clinical utility of ctDNA-based MRD assessments? One thing that may help us in the near term is to help us adjudicate PET samples. So for example, patients with residual uptake at end of treatment, we could determine if there's a high risk of recurrence, particularly in borderline levels of FTG uptake where a biopsy cannot be easily obtained. And then we also might be able to identify relapse earlier. However, the clinical significance of early identification of relapse is not clear at this time. I think one thing that's really important as an oncologist at an academic center is that any MRD-based test that is utilized in clinical use has to not just have excellent performance for prognosis, but also clinical utility, which essentially means once we have a result, an oncologist will need to know what to do with that result, not just have it for just for the sake of having it. So for example, well-designed studies knowing what the biomarker means and having some intervention that is able to reduce the risk of having the positive biomarker. And so this is called out at the beginning of this webinar. We showed some of the discordant results of the use of ctDNA as a colon cancer biomarker, because while multiple studies continue to show post-operatively in colon cancer that ctDNA is associated with worse prognosis, it's hard to know what we need to do about it at this time. And so that's why the NCCN just earlier this year does not back the use of these now more and more available assays outside of a clinical trial.
All right, thank you to all of our panelists for their data rounds discussions. To summarize here, before we move into our Q&A, today we've discussed that phased-seq is superior to PET at end of therapy and also predicts remission at earlier time points during treatment. We've also discussed that phased-seq has shown concordance with progression-free survival in a clinical trial and disease recurrence up to 12 months earlier. And although there are many important considerations around MRD testing broadly, this demonstrates the potential utility of an ultrasensitive MRD test to improve clinical care and at the end of therapy time points, as well as in a trial setting, to potentially accelerate new drug development. All right, and with that, we will move into our Q&A session. As a reminder, please use the Q&A feature at the bottom of the screen to submit any questions. Please do not use the chat feature. If you include your name with the question, we may be able to follow up with you following the webinar. If we run out of time to answer all the questions today, you also have the option to enter questions anonymously. And as a reminder, we are recording the webinar, so if you missed any part of it, we'll share the recording in a few days. So with that, we will shift into our first questions. So I'm going to direct this one initially to Ryan. We have a question about thoughts on the status updates for the Lugano classification revision during last year's International Conference on Malignant Lymphoma, where it was mentioned that ctDNA was not yet ready to be included in the next response assessment.
Yeah, thanks so much. I've heard lots of people's opinions about this. I attended the meeting, heard Dr. Chesson's discussion at the end of the workshop, as well as other thought leaders' opinions on this. I wonder if one of the challenges in this is the lack of a defined MRD product or tool, as well as a defined intervention for what we should do about it. Sort of what I was alluding to at the end. There are a variety of different products out there, and it's hard. The performance is not the same. So I think in that respect, I can agree that it's not ready because there simply isn't a consensus around what tool we should use and what it means. And so I think the development of that data would make it a lot easier for specific settings to integrate it into the response criteria instead of here it is in the response criteria for all lymphomas. It could be sort of directed for specific situations.
Great, thank you. I think in a similar vein, this question is in reference to ALPHA3, but I think we can answer it more broadly. I'm going to direct this one to Dave. Considering the prognostic capability of ctDNA and lymphoma, it seems like an equipoise issue to rank DNA positive patients to observation only.
And that's a great question, and one that we really think about a lot. I think it goes along with what is a standard response assessment. I love the optimism of the question that already is forward thinking about ctDNA potentially replacing a standard of care PET-CT scan. I am also a ctDNA optimist, but I also as a physician, you want to be careful with some of these decisions before we jump in with both feet. I think that no test is perfect, and there are individual examples of patients who are MRD positive at the end of therapy who have not yet relapsed, right? And I think that's the way I would phrase it, is that they have not relapsed yet. So I think without more data from a lot more patients at the end of therapy, it's a little bit premature to say that ctDNA is destiny. And I think that's also what Dr. Chesson was getting at with the ICML response classification. I think we'll get there, but I think that at this point, if it's not part of standard response criteria, it's a little bit hard to say that the standard of care for a ctDNA positive patient should not be observation.
Okay, great. We have another question. I'm going to direct this to Dave again, though others feel free to jump in. Can you give any information on indications that seem to be problematic, showing prognostic or clinical value other than colorectal?
Yeah, it's a great question. So I think that it's a matter of how low you need to go for MRD and the performance of the tests. I think lung cancer is one that has been problematic, particularly at these key post-surgical landmarks. ctDNA levels in lung cancer are low. ctDNA levels, I think breast cancer is another one, are low. I think we also need to tease apart. We need to tease apart a landmark detection versus serial surveillance. A lot of times these get lumped together in the manuscripts that you see using MRD literature. Serial surveillance is almost always prognostic, right? I mean, if you follow patients, you almost always are going to find that lead time for detection prior to clinical relapse. That lead time may be short depending on the assay, but you'll largely see that. I think the question of at the key post-surgical landmark, or at the key curative intent landmark, when we as providers and we as clinicians want to make a treatment decision about adjuvant therapy or additional consolidation therapy, that's where we see tests have modest performance so far with the current tests available. I think that's true across a range of cancers.
Great, thank you. Lara, I'm going to direct this next one to you. Based on the results presented, are there plans for BMS to implement MRD prospectively in clinical trials?
Yeah, that's a great question. And there is a great deal of interest in ctDNA profiling and how we can apply it to our research and clinical studies. These are ongoing discussions we're having internally about what our next steps are. I think to move forward using this as an endpoint, we need robust data sets that give us the confidence in those next steps. We are talking about how we can apply ctDNA to support efficacy endpoints and biomarker data, and how ctDNA can be used to accelerate timelines and enable quicker decision-making.
Great. I'm going to direct this one. I think all of you could answer this. Maybe I'll start with Ryan. Where does the panel see phased variant-based MRD being the most relevant?
That's a really good question. I mean, I guess, I think we to discuss some of the early data that is seen here. I think one of the things looking at the performance of it, the negative predicted value is pretty remarkable for the product, for phased variants compared to everything else that's out there. So I hope that in two ways. One is I hope that studies in the future can be designed to try to reduce treatment burden in patients who may be cured far earlier than we anticipated. And I think that data is being generated now. And then the second way, and this is certainly part of the Allogene study, which you all have announced in your collaboration with Allogene, is for patients who are destined to relapse, finding an intervention for them. And so I think, unfortunately, PET-CT is so widely available and is so established, and it does take a lot of time to get the data in place to try to support these things. But it sounds like some of these are ongoing, which is very exciting.
Yeah, I'll jump in on that one as well. And actually, I'll give two answers, right? So one is, I think, one that Ryan said. I'm going to echo that. When I was in training for a fellowship, it never made sense to me as to why every patient with large B-cell lymphoma needed 6 cycles of R-CHOP. That made 6 cycles of polar R-CHOP if you're a Polaris person. I don't understand that. That does not make sense from first principles. So I would love to be able to move away to give the right patients the right amount of therapy. I'll also turn it back to the people who are attending the webinar and say, I think it's hard to say without novel clinical trials that come from either KOLs or come from industry collaborators that try to move the needle forward. So I think it's yet to be defined. I think large B-cell lymphoma, of course, is a great indication. I think that we've seen early signs that Hodgkin lymphoma and follicular lymphoma are also very phase-seq amenable. And this is why we need studies to show what the most promising opportunities are going to be to realize that personalized therapy approach.
Great. We do have a similar question, I think, around the future state of our MRT utility in lymphoma, if we expect it to be most informative after end of therapy, or if more of a surveillance monitoring that requires frequent testing, and what time points you would expect yearly, every 6 months, for optimal surveillance based on the data we've seen so far. Maybe, Ryan, you can kick us off with this one as well.
Yeah, and this is one where it may go against the grain of some others. I'm not sure, as David mentioned, just because of lead time bias, the role of surveillance in somebody who's already achieved negativity. I'm not sure what the role of that is, because I think, especially for phase-seq so far, that negative predicted value is so high, it's hard to justify doing serial testing in those patients. There could be ways to adjudicate those positive patients at the end. So if someone's positive at the end, what we don't know is how many of those would convert 6-12. So the data set we generated with NCI, because of the way these studies were banked, we can't just go to some time point with this patient and say, what was the status? We just don't have that. So it's hard to know. Maybe it just took longer for those patients to clear and the subset that didn't. So I think surveillance may not have as much of a role unless we clearly define that patient population.
Okay. A question for Laura here. There's a question about the TRANSFORM trial. Are you surprised that PR patients have similar levels of ctDNA as CR patients at month 3 and the subsequent time point?
Yeah, we're still trying to understand the partial responders in our study. We do not, that's a caveat with our study. Our study was very successful. Most patients tend to be in complete response. We don't have very many partial responders. We do see that patients with partial response begin to clear ctDNA. They just don't seem to achieve that same level of negativity that the complete responders achieve. So it really comes down to achieving undetectable ctDNA status is strongly correlated with positive, favorable clinical outcome.
Great. I'm going to throw this next question to Dave. Given the long turnaround time for results, how do you envision this technology being used as a tool in the clinic? What does it mean for potential future use in heterogeneous and aggressive diseases such as AML?
It's a great question. So a large part of the effort in terms of getting this assay into the clinic is building the infrastructure for being able to do real-time reporting, being able to do not just retrospective data, which the Rashkowski data set that's been looked at, the Sampson data set, those are all done on banked samples that were planned and being analyzed and done together. The infrastructure at Foresight is going to allow real-time reporting similar to other genomic tests that you have in the clinic today, right? Foundation Medicine has their Foundation Liquid assay. I think they turn it around in 7 to 10 days. Our goal is less than a 2-week turnaround time at Foresight for the real-time assay. I think that time is reasonable for in patients with large B-cell lymphoma relapsing after the end of therapy who are in a PET CR, they don't relapse like tomorrow. They don't relapse the next day. So I think that time is reasonable. AML and other hematologic neoplasms are different, right? And they will take different approaches and different assays. So phase-seq, just to be clear, this is the off-the-shelf panel is a B-cell lymphoma or a B-cell malignancy platform. AML is going to take a different approach. And I think that the performance characteristics that you want on a disease that relapses as acutely as AML is going to be different.
Great, thank you. I'm going to send this next question to you as well. Are there any plans for the ctDNA assay based on phased-seq to be approved by the FDA for use in the community?
Yeah, so I think that this is what we need to move towards, right? This is what we need to move towards, and part of why when I first started working in this field in 2014, wasn't phase-seq at that point, but we thought we'd have ctDNA and I'll be using it in the clinic in like 2 years. It's a game of inches, and we move forward year over year. And I think that this is honestly part of what Dr. Chesman was talking about with the response criteria, right? I think that not having an assay that's widely available for routine practice in the clinic is part of that reason. We need to build the data. That data is growing. You're seeing some of that data here today. But in the long run, I would say, yes, that is the plan, right? The vision is that this is something that should be available for every patient and every provider. That's on the roadmap. And the question is, how do we get there and when? And I think studies like ALPHA3 and this type of data that we're generating now is getting on the way to that roadmap. I would want to make it so it's a clinically meaningful assay. I mean, that's really important, right? We don't necessarily want an assay that just is another bit of data that providers and patients don't know what to do with. I think that's why these studies are so critical to get first before we go and look for that regulatory approval to be in every clinic in the world.
Okay, we are coming up on time, so I'm going to pose one last question. It's a two-parter though, so I'm going to give the first part to Lara and the second part to Ryan, but I'll ask you to try to keep your responses brief. With current therapies in development have a pretty similar response rates in non-Hodgkin lymphoma. Do you think this assay has the ability to differentiate therapeutic arms, regimens, or therapies from each other? And then the second part will be how likely would you as a physician leverage a therapy with better MRD data to select one over the other? So Lara, let's start with you.
Yeah, thanks. I think this is a great question. And as a researcher, I do especially as someone that's in early research as well as late stage research, I do think that ctDNA can help us make decisions on assets to differentiate assets from one another. But I also think that when you look at the big picture of ctDNA monitoring, one of our goals is to verify, to confirm the accuracy of ctDNA with overall response. But I do think that we will be able to differentiate assets and finds those that are driving the best response rate for the patients using ctDNA. Ryan?
Yeah. I mean, I think if you have two equivalent treatments with similar toxicity profiles, and one you can use MRD data to reduce the treatment burden on a patient, that would be very compelling, for example. Or the flip of it is where you had something where it's sort of approved concurrently and you could identify early treatment failure, that would be relevant. But I think certainly you have to do the balance of toxicity of the therapy. But yes, it would be compelling to have MRD as a tool to help guide therapy to patients.
All right, with that, we are out of time for today. So thank you again to all of our panelists, and apologies to anyone who submitted questions that we ran out of time for. As a reminder, you'll be notified by email in a few days when the webinar recording is ready to watch, if you'd like to rewatch any portions or share with colleagues. And thank you all for your participation today.